Hello and welcome to another video from MrWatkins.com. This one is going to be on the cell cycle. So let's go ahead and get started here. The cell cycle is a process that the cell basically uses for growth and for the organism would use for repair. So this is for growth and repair. Now here's the thing, if you get injured, cells die. You get cut, cells die. Those cells have to be replaced. Well, how does that happen? Cell cycle, that process of making more of those cells, is what occurs. Cells also may divide because of their sheer size. A cell can grow only so big, and then the volume and the surface area don't line up and allow for diffusion. And you also have another problem with the size of a cell. If it gets too big, the amount of DNA that's found in the nucleus that has to be copied in that process of transcription so that translation can occur and we have proteins made. That process, you just got to have enough DNA so that you can get all the proteins you need all made. So the cell cycle, we're going to look at it as divided into three pieces. The first piece we're going to look at is called interphase. An interphase has has three phases or three pieces in it as well. It actually has four, but we're going to look at these three. And the first one is called gap 1 or G1. And this particular part is the part that the cell is basically doing metabolism. Sometimes interphase is called the resting phase, and that's because the cell is resting from active division. Now, metabolism, remember, is all the chemical reactions that occur in an organism. So this is things like protein synthesis, making fat, lipids. It's the Golgi apparatus doing its processing of repackaging proteins and fats and getting those things ready. Endocytosis, exocytosis, those things are going on. You're looking at processes such as active transport across the membrane. This is the basic day-to-day needs and chemical reactions that carry for or do the needs for the individual cell. Okay? Now at the end of gap 1, there's a checkpoint, and that checkpoint is there to see if the cell needs to divide. Perhaps it's gotten too big, that we talked about a minute ago, or the cell is going to go into what's called G0 or gap 0. Gap 0 is kind of an offshoot of this cycle. It is an off-ramp, if you will, where the cell goes into this particular phase, and this is where it stays and typically does not go back into the cell cycle to G1 or back into the other parts here, because the cell is just not going to divide. There's a lot of cells in your body that do this. They become adult cells. The stem cell differentiates into that adult cell, and those cells then basically fall into G0, and they live out the rest of their life in just a metabolic processes, taking care of their responsibilities in the tissue that they belong. So let's say that checkpoint, there's a cyclin there and there's kinases there, special proteins that regulate the cell cycle, and they say, no, we're going to go ahead and divide. And at that point, then, in interphase, we go to what's called the S phase. And the S phase is DNA replication. We've studied DNA replication, and this is where that occurs. Now, please note, these new replicated chromosomes have a name, and they are called sister chromatids. So our chromosome count doesn't necessarily change, but the number of chromatids actually does. And we're going to keep those chromosomes being called sister chromatids, those replicated chromosomes, until they actually split later on in this process. So gap 1, metabolism, basic day-to-day stuff. Checkpoint hits, hey, we are going to divide, so we've got to make a copy of the DNA, one copy for each of the new cells or each cell. And then after S phase, then we have gap 2 or G2. And of course, we're going to have some metabolic reactions going. But the other thing that needs to be done here is we need to increase the number of organelles so that when the cell does finally divide, we've got enough organelles to handle the metabolism that needs to occur when this cell is back into gap 1. All right, so that's interphase with its three sub-pieces, just leaving out that G0. The next part is called mitosis. And mitosis is a process that is just nuclear division. So we're just looking at the nucleus dividing. Now you need to understand that it's almost like there's this progression that says, all right, at interphase we're going to start doing these things, especially in gap 2, that we're getting ready for to divide. We're going to increase the amount of energy storage molecules. We're going to start putting some proteins and things into place so that we can build some structures to help with this division that's beginning ready to occur. We're going to double our organelles, or at least replicate several of them, so that when we have those 2 new cells, everything will work out. Then we actually start the process of dividing, and we're going to start with that process basically from the inside and work our way out. What ends up happening is the nucleus needs to divide first. Now in interphase, when we looked at the cell, and I'm going to do that in a little different color just so it stands out a little more, and in interphase, the cell was typically what you saw when we did the cell lab, and you've got your cell here, and I'm just going to do an animal cell, and with its nucleus, and you may or may not have a nucleolus, but that nucleus is fairly grainy, fairly uniform on the inside here. Okay, when we hit into mitosis, it is something different. It looks different, and the first part of mitosis is called prophase. And there's a lot that goes on in prophase. In fact, in mitosis, that part of this cell cycle, prophase is going to be the longest part in that cell cycle. Now, prophase, this is where the nucleolus disappears. This is where in an animal cell, the centrioles move to the opposite poles. And I'm just going to draw a picture here to kind of give you an idea of what's going on. I'm going to draw this cell a little bigger. The nucleolus is gone, so you're not going to see that little dark spot that you might have seen when you did your cheek cells. The nuclear membrane is going to break apart and disappear. So those chromosomes that are sitting in there, they're going to start curling up and becoming a tighter mess so that they can be divided out equally. And so we've got our chromosomes, and what you see in these cells is these chromosomes are all bunched up, and they actually become almost like little fingers, or little granules, or pieces. And it's not granules like in a grain of sand. They're much more almost like little sausages maybe inside the cell. The spindle apparatus, which is a special protein, a special fiber that occurs, that spindle apparatus is going to start, this is an animal cell, is going to start growing and growing towards each one of these chromosomes here. And I'm going to draw another centriole here on the other side. And if you remember, the centrioles were paired, almost tube-like things in animal cells. And if you remember, that was also the centrosome where they were found. And this is where this spindle apparatus that I did in red, and I'm not really liking that color, so I'm going to switch over here to blue. But these spindle fibers then basically grow and develop and become attached to the chromosome. Let's talk quickly about what that chromosome looks like. That chromosome, we usually will draw it at this point as an X. And then here in the very center you have a centromere. That centromere has a spot on it that's called the kinetochore, and this is where, right there on that kinetochore, is where the spindle apparatus attaches. So prophase, we're getting ready for the actual cell division. Now the next step here is going to be metaphase. Meta meaning middle, and basically what ends up happening is these chromosomes now separate out. They line up, actually don't separate out, but they line up on the midline, or on the equator of the cell. So here we go. I didn't pick the same color, but here's our cell. And our chromosomes now are lining themselves up on this equator. And I'm going to go ahead and draw them kind of as an X. So here's our chromosomes right here, all lined up nice and neatly right here in the middle. We still have our spindle fibers all attached to each one, and we have the other end of those spindle fibers down at what used to be the centrosome, but now is where the centrioles are. Now, if this was a plant cell, it would be attached to a special part on the cell wall found inside that plant cell. Metaphase just means they get lining up. Anaphase is the next phase, and this has a very distinctive look to it. But this is where those centromeres, the inner part here, pulls apart, and these sister chromatids now, each one becomes the actual chromosome at this point. So let's go ahead and draw this one out here. So here's our anaphase cell, and you're going to start noticing that it's starting to get a little more oblong. It's not as round as it was before. And we still have our chromosomes, but because the spindle fibers are breaking down, because this centromere and these chromosomes, sister chromatids, are being pulled apart, or excuse me, are separating, you get this almost characteristic V shape to these chromosomes, and it almost seems like they're pointing back in a direction towards where that spindle fiber is going. They're going towards the pole here, and they're not being pulled by these spindle fibers. And that's a real key thing, especially for pre-AP students to kind of understand. They're not being pulled back by spindle fibers. They're actually destroying the spindle fiber, and the spindle fiber is actually shortening as it goes. Think of it almost like a path that these chromosomes now know to take, so that they'll go to the opposite sides of this cell. And the next thing we have, or the next part, is telophase. And telophase is a piece where you're really starting to see the chromosomes. They're at the different poles here. And I'm going to go ahead and draw a little dip in here, but the cell is actually starting the process of dividing out that cytoplasm. And so our chromosomes now are going to uncoil over here. They're basically at the other side, at the other pole. We're not really seeing a lot of the spindle fiber anymore. If you do see it, it's very, very short here, pointing right towards that centriole that's there on the end. So telophase is where the chromosomes are now at their pole. Now, if you notice here, I'm going to draw a little line there, and we're going to talk about this thing called cytokinesis. Cytokinesis is the process by which the cytoplasm divides, and that actually starts before telophase ends. And so even though mitosis hasn't quite finished, cytokinesis continues or begins, and this is where the cell cytoplasm divides, the organelles are divided up, so that you end up at the very end of all of this really having two new cells. And so this, in a plant cell, a cell plate will form, but you're basically ending up getting two new cells, and you have your nuclear membrane reform. The chromosomes have all unwound, and we're now back to where we were at the very beginning here, back up at gap 1 with the beginning of the cell cycle. This is the process. Now let me show you a couple of pictures here so that we kind of can get an idea of what's going on. Now here's some pictures of some actual cells. I'm going to zoom in real quick so that you can see those. And here we go. This cell is a white fish. It's a type of fish. It's an egg cell, or excuse me, it's a cell pulled out of the fish. And this is kind of what interphase will look like. And then here you can kind of see in prophase, where we've got the chromosomes are kind of bundled up and they're starting to form a distinct shape. There's no nuclear membrane around it. Then you look here and you see all these chromosomes lining up on the midline here, just like I showed you with this particular one, getting ready to divide. And it may be very hard to see, but there's actually, you can almost see little lines here. Here we see the anaphase, where the chromosomes are working their way back towards each of the poles. And then here we've got cytokinesis, this pinching in that's occurring. In plant cells, you're going to see a cell wall, and I'll show you that picture here. But the chromosomes are almost all the way at the other pole, and so we're getting ready to go through the final process, which is cytokinesis. Now, this top piece here, this other piece, is a plant cell. I believe it's onion root tip. Here is a interphase cell, pretty big nucleolus, but we've got there may be some stain issues there. Now, notice here. We've got a little bit of this nuclear membrane. This is probably early prophase, but you can see we're starting to get, instead of a nice grainy look, we're getting a bunching or a clumping up of the DNA. Here we've got that distinctive line. You can just see a line runs right across it. That's that metaphase plate, that equator on that cell where the chromosomes are lining up, so those, or sister chromatids are lining up so those things can separate. And then here you can see it's very characteristic. You've got this trailing arms of the chromatids trailing back away from the two poles on this cell. And then here you may be able to see it, but there's this cell plate in this plant cell that's lining up. You can see the congealing of the chromosomes as they kind of come together. We're going to form that cell plate, or excuse me, that we're going to form that nuclear membrane. This picture is also used quite a lot. Let me scroll back out here real quick. This picture is used quite a lot, not in just AP, but also in IB. And I wanted to kind of go through this picture with you really quick. So this first thing that you see here, A, this is gap 1. B is going to be S, and C is gap 2. So this whole process right here, this whole process here is interphase. This whole process here is going to be mitosis. And then here is cytokinesis. Now the first part, remember, in mitosis is prophase. And then we have metaphase. And then we have anaphase. And then telophase. And of course you have your cytokinesis. This is the actual cell division part. Okay, so D is going to actually be that cell division, where A, B, and C are going to be your interphase part, with A being gap 1, B being the S phase where the DNA is replicated, G being gap 2, and then we start our process of cell division, divided up between the nucleus dividing, mitosis, and then the cytoplasm, the organelles dividing, which is cytokinesis. That's it for today.